Okay, so in the last video we were done till the type of cofactors and their work. But in today's video we are going to look into enzyme inhibition, or we can also call this inhibition as inhibitors. So what are these? Like the substance which inhibit simply means reduce. The substance which inhibit or reduce activity of enzymes are called enzyme inhibitors. Okay, enzyme inhibitors, I'm just writing like this. So like why they are required, or why they are supposed to inhibit or stop or reduce the activity of enzyme? Just because in sometimes enzyme does not work very properly, like they do not work properly, or they do not run the system they are supposed to do that. So enzymes are supposed to reduce at that time. The activity of enzyme, it has to be decreased, because at the time the products are like they enhance, or the products are getting too much but they do not require it. For that they need to inhibit or reduce or stop the activity of enzyme. So in we have three types. What are the types? First one is competitive inhibitors, just I'm writing like this. Second one is non-competitive, and third one is uncompetitive. This uncompetitive is really not very much important, but we need to discuss to know the concept, like what's the difference and these all. So in today's video we are going to discuss the competitive inhibitors. So what are the competitive inhibitors? So these competitive inhibitors are similar to the substrate. So if we have here, the upper one part is known as substrate, this one is enzyme, the downward part is enzyme, but the side, like this one area where they get attached, is known as active site. Okay, so now like competitors, like this means simply competitive, this means they have the competition. When we have studied, like if any one of us gets the highest marks or anything, so this means they have the competition between. So whenever if the competitors would be more, so the competition would be more. So if we have the competitor more, so substrate would be also more. So now question is which will be attached on this one side, like what will attach on this active side. So in these both, whatever will be in the more concentration or the more quantity, that will be attached with this active side. So here the competitive attached with the active side, but they were not supposed, like if we have this the competitive, this will be attached, but the substrate was supposed to attach with that or bind to with the active side. Instead the competitor was attached. Okay, so why just that? Just because maybe at the time the concentration of competitor would be more, just because of that. Then if we just talk about the example of this one, so first let me write the definition of this first. This means they competes, or they have the competition between with what? Competes with substrate. Compete with substrate. And for what? For the active site, for a site. Okay, active site, as I just told you, the part where they got attached is the active site. So there is a competition between where the substrate will be attached or the competitive would be attached. So the example are metronate, we're not supposed to discuss these, and another would be sulfonite, and we have another bacteria which we are discussing. In that no reaction